So let me introduce you to Project Lamport. Project Lamport's a trial site that's been running for about 12 years now. It's about 8 miles north of Northampton. It's on some heavy silty clay loam soils, and it traditionally was set up to look at cultural ways of controlling blackgrass. And of course, one of the ways we can do that is to move to spring cropping. And of course, with spring cropping comes the opportunity to include cover crops. So not only did we have that control of blackgrass, but also we started to see the health of our soil, and particularly the structure of our soil, starting to improve as well. I've been involved about 6 years looking at kind of a more science approach, a replicated work, and trying to really dig in quite deep into soil health, into cover crops, into blackgrass control, and a number of other things as well. And there's some unique opportunities that we've got at Project Lamport, and as I mentioned, one of them is grass weed control, and that's been done a number of different ways, but rotationally, but also using spring cropping and cover cropping as well. I'll come back to that in a moment. And with cover cropping has come the real opportunity for improving the health of our soil, and we often talk about the way the soil functions. I'll cover those details in a moment. And again, cover crops have played a very significant role in that. And the cover crops also have enabled us to get energy into the system, and that's a critically important aspect whenever we're thinking about our production system or rotation. Any opportunity to have this unique carbon pathway of using plants, and in our case cover crops, to get carbon or energy into the system. We've tried extensively to remove as much intensive tillage, and I think we can say that we've removed all intensive tillage. Have we been able to remove entirely tillage? I'll come back to details on that in a moment, but by and large that's been removed. And at the bottom line, it's getting back into those first weeks, early establishment of those first weeks, with the minimal risk of obviously blackgrass control. And that's been certainly a very successful program. But of course, comes opportunities, comes challenges, and residue management in a no-till system. Much of it we're doing here is a no-till system is always a challenge. But cover crop timings, particularly early establishment and early termination dates, are critical pinch points, if you like, to the success of a cover crop. So if you kind of step over those and ignore those, then cover crops can bite you. As I mentioned a moment, the one little challenge we have is surface compaction. This is compaction around about 8 centimeters. I'll come back to details on that and how we've tackled that, particularly in the no-till systems. And then at the end of the day, it's about attention to detail. It's about reducing axle loadings, tire pressures, machinery settings, timing. Sometimes you come back in the afternoon and drill the crop. In the spring, it's there's a lot less damage to the soil than drilling it first thing in the morning. And at the end of the day, we've all had to cope with shifting weather patterns. But what we have noticed is improving the health of our soil, reducing the amount of disturbance in our soil. Our soils have become a lot more resilient to these large rainfall events that we've been having in the recent times. So at Project Lamport, when we think about soil health, we think about the way soil functions. And of course, we want soils to produce good cash crops and good cover crops. We want them to cycle and manage water throughout the year, both in dry periods and wet periods. I'll come back to that. We want them to cycle, sequester, but very importantly, store. Those words are slightly different. Carbon in our soils. We want a production system that leaves carbon behind, as well as we want to cycle and store effectively the nutrition within that soil. Of course, we want to be supportive and resilient to machinery, especially when soils get a little bit more vulnerable. It's heavy soils and wet soils. They can become damaged very, very quickly. And we want to manage these gases exchange. We want to get air into our soil. Of course, we want to get CO2 and things like ethylene back out of our soil. So we need to have good soil structure to manage those gases exchange. And my firm belief is that most of those functions in our soil are as a result of getting energy. And when I mean energy, I mean carbon back into the system. So what are some of the challenges that we face, these anthropological, these human effects that we face to challenge? Well, at Lamport, and as across the world, they kind of broadly fall into 3 categories. Probably the biggest one is loss of organic matter. And we think we've about 40% loss in organic matter at Lamport. If we measure the field margin, it's about 40% more organic matter than the bulk of the field. So that's our estimation. It's about soil compaction. You see it so often, especially after a wet season, heavy soil types. But for us, we're on very low axle weights now, very low tire pressures. We've not got compaction at depth in our soils. What we do suffer from is this very shallow surface compaction. I'll come back to that later in the presentation. And of course, with these changing weather patterns, these big rainfall events, soil erosion is never far away, and we're on a sloping site. And what's been interesting to us is where we have high tillage systems with no cover crops compared to our no-till systems with cover crops, how substantially different our soil erosion and rills and gullies are forming in those plots. So how do we deal with these anthropological, these human effects? So these are some of the lessons that we've learned: is build systems that leave carbon behind. That's obviously, we want to increase the organic matter in our soils, and we've been very successful at doing that. I'll share more of that later. And reducing losses, and that comes primarily from oxidization, which is a function of tillage. So that's how we've dealt with loss of organic matter. And then compaction, it's as I mentioned a moment ago, it's about having low pressure systems, low axle weights, controlling traffic. Timing's often very critical in that. Reducing cultivation intensity, so improving the strength and resilience of our soils. Timing, avoiding when they're really wet. Going in the afternoon sometimes makes a huge difference than drilling in the morning, for example. And then just managing very carefully this top 8 centimetre profile of the soil with any disturbance that we might need. And of course, erosion is, controlled by, having good roots and good plants growing in our soils, building carbon structure that increases infiltration rates and reducing cultivation intensity. And, at the end of the day, throughout all of those, cover crops play or have played a very significant role. So the Lamport journey is a journey. It started off by looking at grass weeds and reducing soil disturbance, and then improving our soil health. So that's the journey we've been on, and it's about energy, fundamentally about getting energy into our soils, and energy in. Work is done. It's just a very simple principle, really, and functions are performed, as I've tried to illustrate already. So, of course, energy in relies on this process called photosynthesis, and that it ends up storing these sugars, these results of that chemical process, in the shoots, the above ground biomass, in the roots, the below ground biomass, and of course, much of it feeds a plethora of microbiology and macrobiology and all sorts of things in our soils. Come back to that in a minute. So that's the way energy flows into our soil. That's the way we sequester carbon from the atmosphere. But of course, some of it leaves through respiration. It's a living, breathing cycle. And but what we want to do is reduce the losses and increase the sequestration, and therefore provide storage. And one of the critical important things is the surface of the soil. We often travel on it extensively. We move it extensively. We probably don't respect it the way it is, because this is the place where these gaseous exchanges take place, and we have to be really careful that we look after the soil surface. So I often think about soil health, and I'm sure we all do, in terms of biological, physical, and the chemical process. But actually, the more I study it, the more I read papers, the more I measure things at Lamport, then I think it more of an electrical system. And our leaves on our plants are the solar panels, and we need to maximize those, diversify those. We need to do anything we can within a rotation to maximize our solar panels. Of course, the soil is our battery, and some soils will have a big battery, higher clay content soils, for example. Some will have smaller. The number of compartments in our battery is the stable carbon element of our soil. And then the kind of interesting thing is the biology, the accessible carbon and biological attribute is the charge level. And again, as I made a note there, soil structure is really critical to these processes. And then I guess more emerging science, we're still trying to work this one out at Project Lamport, but we're beginning to get a handle on it. There's this whole concept of the redox scale, the EH scale, and the pH scale. And I won't go into too much detail, but suffice it to say, EH is the exchange of electrons, pH is the exchange of protons, and one divided by the other essentially becomes the charge of the soil. And of course, more importantly, from a practical point of view, cover crops and no tillage play a big role in each one of these aspects. And you'll see the data shortly, but we can increase biological activity with increased stable carbon. If you like, although it's difficult to measure, we've optimized EH, we've buffered pH, definitely, and no-till reduces this, reduces oxidation, which is essentially energy leaving the system. And very important to this whole process, we've improved our soil structure, which is a critical element to the success of this, getting energy into our soils. So energy in, work done, functions performed is what we're looking at. And this is the big chemical process. I won't go into too much details, other to say that soil structure is critical. And reason it's critical is we need air in our soils, we need to manage water in our soils, and without really good soil structure, those things aren't going to happen, and we are not going to store carbon successfully unless we have really good soil structure. Of course, minerals are important. I'll come back to that as well in the storage and capture of carbon. So we've been busy collecting lots of things. I'm not going to go through all of these, but these are all our significant results over the last 6 years through our replicated work. And for every one that's not significant, I can assure you there's many that aren't. But it's been a really exciting journey, and I want to just share one or two of these kind of aspects with you, really. So what are some of the key cover crop and catch crop benefits? Well, it's about feeding these guys. It's about looking after all this plethora of soil life, because they bring function to our soil. They bring structure and aggregation, workability, trafficability, tilth. They decompose the organic matter and provide that long-term stable carbon that we want in our soils. They provide nutrition through symbiotic relationships with fungi and bacteria, cycle nutrition. They fix nitrogen. And of course, they play a huge role in the water holding capacity, drainage, and infiltration, as you'll see. But very important, their food source is this photosynthetically derived carbon. It's a unique pathway. We can bring organic material onto a field from where it's been sequestered somewhere else, but only plants can sequester that carbon in that place. So earthworms, we started looking at earthworms very early on in our work. You know much about earthworms. I won't go into too much detail. You can see the big functional groups here. We've got majority of our earthworms are unique earthworms. These are deep burrowing earthworms, which is what we want. They provide the burrowing, the porosity into our soils. And so this is our data over 5 years, extensive piece of work. And we started in year 1. We do this work in the spring of every year, so spring 19. Cover crops made no real difference. We had big, blocky, poor structured soils, and but tillage actually. The left-hand 2 bar plots are our controls. There's no cover crops in those, and all the green ones are cover crops. But the darker bar plots are the ones that had tillage, and you can see that tillage pulled earthworms down in the first year. And indeed in the second year as well. But in the second year, you can see cover crops, if you look above the red line, significantly increased worse, and that continued in the 3rd, 4th, and 5th year. And the 5th year was lower, but if you remember, the summer of 2022 was extremely hot, and we suspect the lower number as a result of that weather change, if you like. But nevertheless, cover crops are clearly significantly increasing our earthworm population. And you know, what's been their energy? What's been an energy? Well, it's been the carbon, it's been the cover crops, it's been the residue. It's that kind of mix of things that provide energy for earthworms. And you can see here our cover crop systems, the green ones, have significantly higher worm numbers compared to our control plots. And we've measured cover crop biomass from NDVI data here. But that's great. I hear many people say, well, that's great. It's a sign of life. Are they worth having, and do they provide any function? Well, globally, there's been some really big studies recently published where, you know, if we removed our earthworms, they have about a 6.5% contribution to global grain yield, and that rises to about 8%. And much of that is coming from nutrient cycling. So that's probably one of the biggest functions that earthworms perform. But for us at Lamport, we've looked at this function of earthworms, and it's about these channels in our soils. And this is from a HDB monitor farm in near Norwich, but you can see that, you know, I'm always amazed by these burrows, the dark carbon around them, the way the roots channel through them. And of course, that's where the water will channel as well. And for us, it's been about infiltration rates. So this is a structure we started off with back in 2018, 19. This is the structure we've ended up with in more recent days. And you can see, you know, this is laborious work, but you can see in the first 2 years, looking at infiltration rates, and each one of those bar plots is 24 separate readings. You know, we saw no changes in earthworms for 2 years. And then in the 3rd year, we began to see cover crops just starting to significantly increase infiltration rates. And then it's almost got exponential in the 4th year. You can see huge increases in infiltration rates. So biology works, structure drives this, but it takes time. It doesn't, some of this stuff doesn't happen, and there's patience involved. It doesn't happen overnight. But again, it's about energy in, work done, and of course functions performed. The function is infiltration rates. And as you can see from these 2 regressions, you can see that where cover crops have significantly increased our infiltration rates, and worms are, you know, one of the key drivers for that. Of course, we've got channels created by roots and all sorts of things going on, so better structure, but worms play a big role. So do worms bring function to our soil? They bring nutrient cycling we've seen, but also infiltration rates as well. But plants are quite selfish, and they invest in carbon, and they do it because of this kind of evolutionary concept we call extended root phenotype. What they're trying to do is when you bring plants and soils together, 1 and 1 doesn't make 2, 1 and 1 makes multiplication, and they bring many different extended phenotype to thing. And the one we've been looking at is soil health and soil aggregation. And we know, I think most of you who played around with cover crops will know that when you have a cover crop going in the field, you start digging underneath that plant, the structure of that soil is significantly better. And that's one example of an extended root phenotype. So only plants and biology working together is holobionts. A holobionts is a discrete ecological unit with other species around it, can produce very fertile, well-structured soils. And soils have structure across many different spatial scales. So I put a scale there, but I haven't put any units on it, because whether you look at it in a spadeful like we are there, the structure looks great. You've got lots of cracking, lots of small pores, big pores, you've got worms in there. But if you put it under a microscope or even through an X-ray machine, you can see that structure is the same, whatever kind of magnification level you think. And it's one of the unique materials in the world that has the same structure across a wide range of spatial scales. It's a fascinating medium. And of course, all this biology that I've been talking about has different functions in a soil. We've talked about earthworms, but we've got fungi that glues things together, sews things together. We've got bacteria that sends out, glues and paint things and eat things. There's a myriad of different roles, biotic roles, that create this fantastic soil structure. And one of the fascinating things it does is the way it structures the soil and manages moisture content. So matrix potential, if you've never come across this before, is the ability for a plant to suck moisture out of the soil. It's a negative pressure, and moisture content obviously is the volumetric moisture content within the soil. And what we want is a wide range of pore sizes. So we want big pores, so that right at the top here, when we've had a rainfall event, soil drains very, very quickly. And then we want lots of small pores down here, so it holds on to the moisture when moisture content declines. And only biology can do that. Tillage will give you lots of big pores, and in the very short term, we might drain soil. But as we know, when we get a big rainfall event again on top of tillage, it often collapses that structure. We want this biological structure that gives us a wide range of spatial scales. So that's called the soil release curve, and biology is really, really important to that function. And again, it's driven by carbon. And of course, all these pores, we want them connected together, because only when they're connected together do roots go down there, does moisture go down there. We don't want localized pockets of good structure. We want structure across the whole profile of that soil. So this is where we've kind of ended up with our soil structure. On the far right, no-till cover crops. This is our control plot, if you like. We've ended up going from very big, blocky, angular soil to start with to this kind of, it breaks down very easily in your hand. I wouldn't call it aggregated soil. It's still a kind of unbiological soil, but just by leaving it alone, it's never had a cover crop in it now for 6 years. It's structuring quite nicely. Next one is heavy tillage, 25 centimeters, pretty much farm standard in this part of the world. Never had a cover crop in, and I think you can see that soil. You can pick up one of those lumps, throw it on the ground. It just doesn't shatter at all. It's a very ugly structure now, and that's we've used tillage when we don't need to. It's an experiment. When you use tillage and you don't need to, that's kind of where you end up, really. The next one, the middle one, is tillage and cover crops, and you can see cover crops had a really good attempt at improving that structure, but of course further down where the roots haven't been, you can see similar damage to our tillage no cover crop. Hopefully you can see some color change, because the next one is no-till. It's got a multi-species cover crops in it. It's had that now for 5 years, no tillage, no soil movement. All of them have the same spring crop. And hopefully you can see that is darker, and indeed it has got more carbon in it. I'll explain that later. And it's got a lovely crumbly aggregated structure. And the last one, the far one on the left, as you can see, is the field margin. We've still got some way to go. But you can see these are all the sort of physical things we're measuring, our VES scores. The higher the VES score, the poorer the structure. You can see the infiltration rates, that's milliliters per minute. And you can see how our bog density is changing as well across those different soil structures. So quite an exciting journey seeing our structure of our soil. And these plots actually are all right next door to each other, with the exception of the field margin. So this is the kind of, I like a little bit of drawing. This is the kind of where we've kind of ended up really after 6 years. So the far right one is our control plot, and that's kind of, nice small plods of soil, break down very easily. But if the soil dries out in the spring, it doesn't happen every year, we're left with this 8 centimeter surface compaction. The second one here is our subsoil plots. It does create a really nice tilth in the top few centimeters, but below, then we're left with this really great big ugly angular wet lumps of soil. And then when we combine cover crops with tillage, we get this kind of nicer structuring in the top of the profile soil. We get worms in there, but further down the profile, we're still left with these big ugly lumps of soil. And the last one, multi-species cover crop, it's darker, we're still more carbon in that, but we're still unfortunately left with this surface compaction when the soils dry out in the spring. It's not every spring, because if you get good rainfall during April and May like we have this year, you won't see that phenomenon, but in a dry spring you most definitely see it. And you see it in lots of soils in the autumn and the spring. Many people have got that challenge. So how do we get there? How do we get from this kind of sort of much lighter, poor carbon, poor structure to this lovely kind of aggregated structure? And for me, primarily it's about photosynthesis. It's the power of photosynthesis. This is a journey we've been on in 4 or 5 years, and you can see the immense changes. It's exactly the same plot in the field, and that's where we started, and that's kind of where we are today. However, if you leave soil alone, as I've mentioned already, no cover crops, no soil movement, it's amazing what it will do on its own. So this is our control plots. We have 4 blocks. We take three digs from each one of those treatments, control treatments. You can see what is very, very consistent. It's not just, I'm not just picking out one tray, they're all our trays. And also the change is also very, very consistent as well. In 3 or 4 years, we saw a huge change, and that continues to improve. So moving on to the work we've done looking at sort of carbon, obviously, you don't need me to tell you the importance of carbon. This is an old hedgerow, and you can see it has a myriad of benefits on our production systems. And carbon is complex, but essentially it's stored in our soils in 2 pools: the particulate or organic matter and a mineral associated organic matter. And it's the mineral associated matter which is the sort of more long-term stable. It's associated with, particularly with clay, and the particulate organic matter can be lost and gained, and it's en route, if you like, to be stored in a mineral way. And we've been looking at lots of things. We've measured organic matter using loss and ignition and DMAS. We've measured it using the CO2 burst, the permanganate oxidized or carbon, sometimes called active carbon, but that kind of definition is changing a bit. Phospholipid fatty acids, microbial biomass, biometer, and a number of other technologies which I'll come back onto later. And cover crops and carbon storage, of all the methods out there to promote increasing carbon there in agricultural soils, this is a paper not too old. Cover crops appear to be the most promising, and that's certainly our experience at Project Lamport. So a few of these things, we've looked at these fatty acid in the membrane of this is a bacterial cell you're looking at there, but we've been looking at this phospholipid fatty acid. There's no other fatty acids apart from what's in the microbial life in our soil. So if we can extract that, then we can measure it, and we can pull out lots of different biomarkers and different biomarkers associated with bacteria or fungi or things like that. And I've managed to identify 45. I clearly haven't put those on one slide. I pulled out the 2 fungi biomarkers, which is saprophytic fungi and the mycorrhizal fungi. And you can see here, compared to our control plots, our PLFA in milligrams per kilogram of soil has nearly doubled in all our cover crop systems. And the last one is field margin. We've still got a long way to get to our levels that we've got in our field margin, but that's been an exciting journey to see that we are genuinely, impacting the microbial life within our soil. We've also looked at it. These are traditional tests, but we've also looked at it in terms of microbial biomass. This is the chlorophyll extraction and fumigation, and so we're measuring the total amount of carbon within the microbial mass and the total amount of nitrogen. And you can see in both of the cases, our cover crop systems are increasing the nitrogen content and the carbon content in our microbial biomass. So 2 very traditional tests, but both strongly suggesting that we're impacting not only structure, infiltration rate, worms, but also the more microbial life in our soils. So we've looked at carbon stock assessments, and just before I go into the results on that one, just a little bit of note about bog density, because it's a really critically important measurement that's probably not always fully understood. But typically in a little ring. These are sort of 50 millimeters by 50 millimeters. The amount of weight in that soil per kilogram cubed is about between about 1.2, 1.5. I think most labs use 1.3 as a kind of baseline. And but it's a critically important part of understanding packing density, and I'll come on to packing density in a minute. And we've measured it at the surface, as you can see. We've measured it at the top 30 centimeters, and we've also measured it right down to 1 meter. So really a lot of work gone into that. And this is a little calculation, and it's often understood, but if you take your organic matter, especially if you're measuring loss and ignition, you times that by 0.58, about half of it is carbon, is what I'm saying, really. So example there, I've given 3%. The depth of the profile you're looking at, in this case 15 centimeters, the bulk density 1.3. If you just do a simple sum of multiplying those together, you get the amount of carbon in tons per hectare. Don't worry about the units, they kind of work themselves out. But in our case, 58.5 tons per hectare. And you can do the same with clay. 30% clay, 15 centimeters times 1.3, about 585 tons of clay. And you can then work out the ratio, which is a really very clever little measurement and gives you a really good idea of where you are in terms of the amount of carbon you're storing in your soil. However, there is a challenge, and it's a quite significant challenge, because not all soils are the same, because you might have a no-till field, which is the one on our left here, and you might have a very heavy subsoil. So we've got these right next to each other in our plots, so we have to deal with this. And because you're moving a lot of soil, you're lifting a lot of soil, you're lowering the bulk density, even though the same amount of soil is in there, but it's over a bigger volume. And the no-till tends to be, a higher bulk density. And if you use the same sampling probe, the same method, you're going to get very different bulk densities. And if you're using that to calculate carbon, you're going to get different carbon levels. And this is where care needs to be taken. And there are methods to calculate this out. I won't go into details, but you need to be super careful when you're comparing one field to another, especially if it's under different management practices. So these are the results of our work. This is down to 1 meter. And when we remove cover crops and we just look at the tillage versus no tillage, over the 5 years, this is over 5 years, we've lost 17 tons of CO2 equivalent. So this is the amount of CO2 leaving our system. This is energy leaving the system. We've lost 17 tons per hectare of that. And so the left-hand plots here are the actual carbon, the right hand are the CO2 equivalents. And look, we've measured this using 96 meter deep cores over 32 plots. There's a huge amount of data in this. And then we looked at the next soil profile down. We saw no change in the top profile with cover crops. We're just looking at cover crops versus no cover crops, so no tillage involved in this. And you can see when we're making that comparison, we've actually drawn down 27 tons of CO2. So we're actually significantly increasing the amount of carbon, but it's deeper in the soil profile. And that's why it's critical to start measuring carbon at least to 30 centimeters, but I would argue probably to 60 centimeters. We've measured it to a meter here. But exciting, we're losing carbon. Not exciting, but we've oxidized, we've moved up the redox scale, we've lost carbon out of the system, we've lost energy out of the system. But further down the soil profile with our cover crops, no-till systems, we've gained carbon. We've come down the redox scale, if you like, and that's been an exciting journey for us. And this is a study in Finland over 20 farms where they looked at all the really limiting factors for carbon sequestration. We've definitely got some of these at Lamport. Looked at it in 3 ways: how to increase photosynthesis, how to increase microbial growth, we've seen that's really important, and how to decrease loss. And I won't go through all of them, but you can see the big pie ones here. The big ones that are limiting is things like poor soil structure, low microbial activity, micronutrient deficiency, and you can see all the others as well. But if we really want to store carbon, we've got to start driving much better soil structure. Of course, these are all linked together. They're not working independently. They're all, microbial life will bring better structure, etc., etc. The really interesting one is sulfur, because half of organic matter is carbon, but the other half is things like sulfur, phosphate, things like that. And sulfur is really important. And Lamport, we really struggle with. The kind of limit we need to get to is about 20 milligrams per kilogram or parts per million, and we're right down at 5. So we definitely cannot. We struggle to get higher levels of sulfur into our soils. And then we looked at soil organic matter in this kind of way of looking at its carbon to clay ratio. And this is the Rothamsted work, where if you're below the red line, you're degraded at 1 to 13. Between the red line and green line, 1 to 10, and the blue line, about 1 to 10. And above that, 1 part of carbon to 8 parts of clay, you're in good. So that's what we define as a healthy soil. So if you take your organic matter, times it by 4.8, if you're using loss and emission, that gives you organic carbon. So that's the example we've got here, 2.61, and I've given you these numbers already, but roughly we've got 55 tonnes of soil organic carbon and 550 tonnes of clay at Lamport. We've now significantly changed that in our no-till cover crop plots, but so you can see our ratio is around about 1 to 10. So we're okay, but we've still got a long way to go. And this is what it looks like through the soil profile. So the blue line is our field margin, and at 0 to 15, subsoiling has significantly increased the soil organic clay ratio. We don't want to do that. We want to decrease it from 9.7 to 1 to 10.4. It might seem much, but a small number, but it's a big effect. The field margin is at 6, just to give an example. And at 15 to 30 centimetres, cover crops have significantly reduced the soil organic clay ratio from 19.3 to 16.1. So we're heading in the right direction, if that makes sense. And again, at that depth, the field margin is 8. So that's what the work we've done with this very important metric, I think, looking at that ratio between carbon and clay. And this is all our kind of same picture as I've shown you before, but this is all our biological tests, and I'm not going to go through all of them, but I've gone through some already. But you can see gradually as I move across the screen how most of them, if not all of them, are improving. Our worm numbers are going up. Our PLFA is going up. That's total PLFA. Our microbial biomass is going up. Our carbon is going up. And look, we've looked at some of these more new emerging technologies, like the work that PES technologists are using to use AI to estimate carbon, and you can see that we've got on very well with that. Microbiometer, we've had to take a lot of samples, but we've made real good use of that technology. Less scientifically accepted, but it's a good little indicator when you're using the field. And of course, we've used respiration, active carbon, which is the permanganate epoxy measurement, and of course, worms I've already shared with you. And then what about yield? So we've had a mixed bag in yields. These are all spring crops, but when we've planted our cover crops in 2018-19, when we planted our cover crops early and we've terminated early, we had a positive effect upon yield. And our mean yield across all the different systems was about 7.5. And rainfall at 42 millimetres in May is critical at Lamport. If we can get more than 40 millimetres, we can get pretty sensible spring crop yields. If we get less than 40 millimetres, you'll see that starts to fall off. The following year, we established late. 2019 was a very wet autumn. I let the cover crops grow longer, thinking that was good for photosynthesis. Terminated at the end of February instead of terminated at the end of January, and we had a big negative effect upon yields. And our mean yields as a result of that were 6.1. Didn't do that again. Following year, again, slightly late. What I mean late is into early September rather than the end of August for us at Lamport. That's the difference between late and early. And we had a neutral effect, but we had 61 millimetres in May, and we had average mean yield of 8.75 tonnes per hectare. Following year, again, a slightly late, early September, but early termination, again neutral, and less rainfall, 5.25 mean yields. And then we moved to spring oats because we want to go into first wheats here next, and so we planted those very early. We had a very dry 2022, earliest disturbance, but again, a neutral. So oats gave us a neutral response. So look, if you're thinking about cover crops are going to be the big yield increase, I think that would be overselling it. It's more about the functional benefits it brings to the soil rather than the yields. However, after 11 replicated harvests of different replicated trials we're running at Lamport, none of our tillage systems have ever once gone to yield in 5 years, which I find quite fascinating. I wouldn't have believed that when I started this work. And then we've looked at what happens in subsequent years after using cover crops. So this is a trial that's now gone. It was in winter wheat. It had 2 years of spring cropping and cover crops. And then we split the trial and looked at different nitrogen rates. So all of these were previously had cover crops in, all these 6 different treatments. I won't go into detail. All of these 6 different treatments didn't have cover crops, some of the tillage, some no tillage. We applied full N to half of them. You can see that had no effect whatsoever. But no N, we saw a really nice, significant increase in yields in our cover crop system. So, that was about a 1.4 tonnes per hectare mean increase. So there could be lots of reasons for that. I've hypothesized that 3 reasons. I've looked at thinking it could be this, whole story around exudation and microbial mediated nutrients going in there. Could it be look at mycorrhizal fungi and all these symbiotic things? It could be the rhizophagy cycle and looking at this kind of bacteria endophytic phase and the free living soil phase. They are just hypotheses, and there could be a million other things. But the exciting thing is that cover crops are having an effect further into the rotation. But when you go to full N, that kind of takes that effect away because it's being supplied artificially. Why do the microbial life need to supply it? Principle. Early establishment is really important. This is a little trial that I've run for a few years, looking at cover crop biomass, dry weight cover crop biomass, looking at 3 species here. I've looked at others, but black oats, 3 planting dates, mid-August, end of August, mid-September. The same with clover, the same with cereal. You can see a big fall off, So early establishment, if we want cover crops to really drive all these functional benefits, early establishment is critically important. End of August is really probably a bit of a cutoff. It certainly is for us at Lamport. And mid-September, very little grows in many of the cover crop varieties. And this has been a really, been a really great find. Really, is the response to cover crops and blackgrass. This is a kind of concept of allelopathy. But the left-hand one is a typical typical cover crop plot. We have big plots. They're 12 metre by 12 meters. And this is a typical control plot. So this is in the year 4. Pictures paint 1,000 words. This is the actual data. So years 1, years 2, and possibly year 3, very, very low blackgrass levels. We've moved to spring cropping. That's really done a lot of good. But just in year 3, we started to see blackgrass creep into our control plots here. And in year 4, our control plots exploded. So blackgrass is the allelopathy effect from cover crops is playing a really significant role in reducing the prevalence of this nasty weed called blackgrass. So cover crops, no tillage, carbon, soil health. For me, it's about flowing energy through the soil, energy for the microbial life. You've seen lots of good things come as a result of that. Does not guarantee increased crop yields. However, small increases are very beneficial for soil health. And these are the kind of main things we've found: more earthworms and biology, better aggregations and structure, obviously carbon storage, much better infiltration, decreased erosion, grass weed control, reduced need for tillage and machinery, much better trafficability, and all around a more sustainable and resilient system. And you've just seen, we're impacting, I would think, nutrition, microbial mediated back into our winter wheat after using cover crops and spring cropping systems. Now, just to finish off, I just want to touch on the role of soil disturbance, because it's something we've extensively looked at, from having sort of farm standard systems here on the left-hand side of my screen, and much more low disturbance, shallow work on the right-hand side of the scene. And so what is this kind of concept of intermediate soil disturbance? What is the optimal level of soil disturbance? And the first thing to say is cover crops. Can cover crops replace tillage? And this is a difficult one to explain, but cover crops are definitely reducing our bog density to similar levels of subsoil plots, but they're not doing it at the surface. They're doing that further down the soil profile. That's 15 to 13. So I'll try to illustrate this using this diagram, but these diagrams are all our different treatments. But if you look at the ones I've just circled with red, these are our no-till cover crops compared to our controls. So the black line on the right is always the same. That's our control plot. But you can see now that our 3 no-till cover crop treatments are reducing bog density at that depth. And if you look at the surface, they're having no effect there. Whereas with the tillage systems, they're the 3 at the lower here, cover crops and tillage, they are definitely lowering bog density at the surface. So the one challenge we got with the cover crops, it's reducing our bog density, improving our structure, but it's not having that effect immediately at the surface. And that causes a challenge. And this is the challenge it causes us. In the spring, if we get very dry weather, as we've had in most of our years, not this year, obviously, 2024, but you can see the no-till systems have significantly less plant counts and vigor than our conventional systems. This has never gone to yield, believe it or not. It's about half the plant count's never gone to yield, but of course we'd want it all to look as it does on the right-hand side. So in March, this is how our soil structure looks in our no-till systems. You wouldn't fault it, it looks lovely. However, the subsoil plots kind of maintain some of that structure, but the no-till plots create this very hard layer. Two things have to happen. One, the soils have to dry out, and two, we have to start with hard soils, and the no-till soils are harder, obviously. And that causes significant challenges. And we've measured that looking at penetrometer data. You can see the black line are all our no-till systems, and the red lines are all our subsoil systems. And so we get this phenomenal structure. Looks great in March. When you get into June, you see a big fall off in plant counts, and it's because of these two phenomena of dry soil and hard soil come together to create this surface compaction. However, the temptation would be to pulverize the soil to death at depth, solve all of that, and that's not the answer, because that brings lots of other challenges, as you've seen already. So the biggest cause, this is for us, the biggest cause of infield variability. And this is a concept I wanted to use called packing density. And packing density is bulk density plus a weighting from the clay fraction and plus a weighting from the silt fraction, because clay and silt play, especially silt, plays a huge role in how the soil's kind of packed together. And what we found is a simple drop of 0.1 of packing density in the top 100 millimeters increases our plant counts by 50%. And I try to illustrate by this little linear regression here. Here's all our tillage systems. These are our no-till systems. The difference between those is a very small amount of packing density. We do not need to pulverize this soil. We just need to stretch it lightly. And so that's where we've ended up with our shallow. We've used a low disturbance subsoil. We haven't got the perfect bit of machinery. We run that about 12 centimeters, which is enough to get enough stretch across the legs, and that resolves our challenge. And of course, that now is part of, or potentially part of, the no-till SFI incentive. Of course, the temptation is to drop packing density by a huge amount, and this is a very large piece of tillage equipment that's just dropping our packing density. I've measured losing a penetrometer, but its packing density has been dropped, and I've kind of caused this mechanical energy in. Of course, we're getting lots of oxidation of carbon as well, biological energy out, damage done. Of course, we're increasing this concept of on the redox scale. And we created a pan at the same time. Of course, over-cultivation causes all sorts of problems to soil structure. Very often at headlands, you get a lot of over-cultivation, drops the infiltration rate. This one is one I saw on Twitter. I thought was very interesting, but it's not used guidance, but there's an overlap as you go up and down the field with a set of discs, and you can see where the overlap is. It's collapsed the structure and therefore prevented infiltration happening. So, over-cultivation, dropping packing density too much, taking the strength of the soil away, is hugely damaging. So this is where I look at soils now. I used to look at it soils, plants and animals. That's the traditional world. But the paradigm shift is now I look at soils always through that lens of photosynthesis, making life from light, feeding our biology, creating structure, improving our plant and the health of our plants. Ultimately, yield is our destiny with this, but improving the function of our soil is probably the biggest benefit we get. So that's really it. That's the journey we've been on at Project Lamport. We have to get energy into our soils through this process of photosynthesis. We have to manage our cover crops very well, early termination, early establishment. We have to manage the top 80 millimeters of our soils. And, we start to do this, then we've seen really significant benefits from the work we've done at Lamport. So thank you for listening. Hope you found that interesting, and